So today's module, and today's and actually tomorrow's, will be related to analysis of gene expression data. And the goal of this part, like for today and tomorrow, will be just basic analysis of gene expression data. So we don't go into much details. So this will we will go up over with Kostya on next week. So we won't be working with like raw data or like R scripts or whatever. And the general, so the idea is to have for you to obtain some general overview of what gene expression data can look like, what you would like to look at it, like what parts you are interested in, and so on. So just general idea. Yeah. So some of this analysis won't be like a perfect one. So again, the perfect ones will be we will be taught with Kostya, but yeah, we'll make it pretty decent ones here today and tomorrow. Yeah, and materials and slides will be available on Google Drive, and yeah, everyone does have access to Google Drive. So there is systems biology folder, and there is day 2, and this presentation is not there yet. Okay, so. Because I would like for everyone to open this presentation, to download and open this presentation, so they can follow it, like go back to the slides and so on. Because we'll have some practice, and sometimes it will be relevant for you to go back. Okay, so the slides should be available for you, for everyone to look at. Yeah, actually, can I make one more thing? Yeah. Okay, so please download the slide, and let's start. So there will be 3 parts. One that will just go over how to look at gene expression data in general, and there will be some like more analysis part and how to work with public data. And this part will be today, and well, people on the Zoom can't see my pointer, but yeah, basically the first part will do today, and the 2 more parts will be done tomorrow. And the pipeline that will, so the overall gene expression analysis pipeline consists like of several stages. One is obtaining of gene expression data, like RNA sequencing or microarrays, and there is some. So we'll work with this public, with middle part, with working with some pre-processed data, and there is also usually when you submit, when you publish your data, your publications, the data that you used, you usually have to submit to some kind of repository. So again, we won't be covering this today and tomorrow. And so basically, what we'll go over like from some kind of quality control to biological interpretation of the data. And like I'll start with a little bit of history. And so when we, like Max told you yesterday, so basically we started doing systems biology workshops back in 2014. And when we did them, we used this software called Genie, and which was very useful. It had this nice heat map interface. So basically here you have like genes and samples and gene expression data nicely visualized, and this was very interactive, and there are a lot of tools there. However, at some point it got deprecated, like 2016 or so. So basically, this was a nice way to show the data to people who like look at it the first time. Yeah, but so this tool was later developed by the same author. This tool was replaced with Morpheus, and Morpheus is a web service that you can load your gene expression data into, or like, load some preloaded datasets and work with it like, nicely in a browser. So it's actually like working simply in the browser, so you don't have to set up anything or whatever. Nice thing that it's while it's web application, the data isn't sent like to any server, so it's on the client side. So it's pretty convenient when you have some private data. However, the problem was that because it was re-implementation and because this was basically written from scratch, so a lot of interesting methods wasn't implemented there. However, because of this because this software was this open source and based on JavaScript, so at some point we came up with the idea that actually you can connect this JavaScript web application with R. And as you probably know, that well, R is one of the most used languages for bioinformatics, especially for gene expression data analysis. And for R, there is a couple of frameworks to make it available through web applications. And well, basically, what we did, we implemented a bridge between R and Morpheus. So we called and created this tool called Phantasus. So basically, now it has the same heat map interface and the client side, client-wise, but actually it allows you to do a lot of analysis methods from R and bioconductive packages. Yeah, so it's also like free and open source, and like we developed this in our lab, and it's still like in development, and like feedback from you or anyone is welcome. And today and tomorrow we'll be working a lot of this, and actually later in the modules we'll be working a lot with this tool. Yeah, but one thing that it's server-side application, so your data is sent to the server. So if you work with some very private data sets, be aware and probably install it on your laptop. Yeah. So again, there's it's a web application. So we have two official mirrors at ITMO and Artemov Lab website. And well, one is in Russian, another in US. And also you can install it locally. So well, at some point at least I used a lot the local version. That's very convenient. But otherwise you can just go there and do some analysis. And actually that's what we'll be doing today and tomorrow and so on. Yeah, so now, well, we talked a bit about the software, but let's go back to the data. And usually there are two ways to obtain data. One is that, well, you do an experiment and get it from your experiment, which costs money. And another way is to find some public data and process. And the public data usually goes from publications. So as I mentioned already, nowadays, like any decent journal requires you to upload your gene expression data to some database, so that like, the research is kind of more reproducible, and that means that you can usually use this data. Well, not usually, but often. Yeah. So, like you mentioned, you have you found some publications that you're for some reason interested in. For example, there is experiment that you are considering to be done, and you're trying to understand will it work, like what changes there will be or whatever. And in this paper, you see like words like gene expression analysis by microarray or by RNA sequencing, which is more probably frequent now. And well, again, this should mean that like if the journal is decent, and this was yes Nature, so pretty decent journal, and so that means that you can go to the methods part, and there you will see some kind of accession number. And there are like two major databases, or like three. but the most common one is gene expression database, and there will be some like accession numbers like this, GSE some number, and well, that's your number that you want to look at. So let's start some practice. So like if you don't know what to do, just Google it. So like let's find out about this data set. Or use DuckDuckGo. And usually, like one of the first links will be the one going to this website. Yeah, and if you scroll a bit, so you'll see some information. So first of all, there will be some abstract about what's going on in this data set. So, well, what happens here, some kind of fraction of the species, some indifferent gamma LPS treatments, and some bone marrow derived macrophages, and so on. So basically, you can try to like have some understanding of what's going on. And yes, you can see here. You can see the link to the publication, so you can actually go back to the publication to check that where this data is coming from. And yeah, as well as some other information, like authors. And now, if you scroll down a bit, you will see this, and click more. So you will see that there are 16 samples in this dataset, biological samples, and from this, well, without like any background, you probably can understand that there is some kind of cell lines or whatever called BMDM, and this is bone marrow derived macrophages as we saw before, and there was 4 biological conditions treated: interferon gamma LPS and interferon gamma plus LPS, and there were 4 replicates for each condition. And just to remind you, so these BMDMs are actually a model of macrophage, a model of resident macrophage on monocyte, and these stimulations like interferon gamma make it activate to pro-inflammatory macrophage. And well, and this picture is from yesterday. Hopefully everyone remembers. Yeah. So there is also like M2 macrophages, which is anti-inflammatory. So it's not here. So and actually you can trying to find some data sets like this IL-4 stimulation or whatever. But yeah, today we'll be working with these M1 macrophages, and similarly to interferon gamma, this LPS also, which is like lipopolysaccharide part of bacteria, which also induces M1 response. Well, when you do it, both of them, you also get some kind of M1 kind of response. So yeah, and so basically you have this dataset page, so where you can find some a lot of information about it. And in general, this database is like one of the major sources of one of the major repositories to put the data into when you do publication, and as well as, well, you probably don't do that much publications, but you can actually read a lot of publications, much more than you have. And actually, you can obtain a lot of data sets from this GR. And this screenshot is, I guess it's not very recent. So actually, let's try to go. To the main website. Yeah, so nowadays there are 150 experiments, 150,000 experiments, so which is a lot. We can multiply it by a couple of $1,000 per experiment. So actually not that much. like $150 million of worth of data, actually probably more. Okay, so this is so now we have our data set. So let's try to look at it. And for this, we'll use our Phantasus. So please open in your web browsers one of these versions, well, either at ITMO or at Voshio. So usually I recommend, like on these workshops done in Russia, I recommend the first mirror, and on workshops in US, the second one. But both should work fine. Yeah. And let's select that we want to open geo dataset, open its number, and hit enter or click open over there. Okay, so at this point, everyone did open the dataset, and it looks like this for you. Yes. Yeah. So if you do have any problems, actually, like for people on the Zoom, I don't see a chat. So if you do have like any problems or questions, please talk about it. Okay. So let me try to. So basically, interface is pretty simple. So you have yeah. So basically, like each of the, so we have this 45,000 rows, which are kind of more or less genes, and we have this our 16 samples. So we can see that there is some annotations, like untreated samples, from gamma treated samples, and so on. And these values here represent gene expression. So like how this particular GOJ7 gene is expressed in like this sample, and you can see some numbers there. Yeah. Well, we can play around with annotations, like you can click show text to show the text, so that it's not just color code. And one major thing here is that the color scheme here is relative. So basically, what like row relative. So basically, what it means is that red corresponds to the highest expression in a row, and blue corresponds to the lowest expression in a row. So, well, these GOJ7 genes have some high expression in LPS and in samples compared to interferon gamma samples. Yeah. One thing that I would like to mention is about microarrays. So actually, like, who is familiar with what microarrays, how they work? Okay. Yeah. So this is like pretty old technology, well, relatively old technology, and which is now getting more replaced with RNA sequencing, but still pretty used, and there are a lot of data for it. And it works in the following way. So basically, we have this microarray, and we have this spot, which has some kind of oligonucleotide planted. And this, like, a short oligonucleotide, like in this case, it's 24-mer. And how this works, basically, you have your mRNA, like messenger RNA, or converted to cDNA, and you basically this cDNA gets hybridized to these probes, and basically, if you have a lot of cDNA hybridized to probe, basically you have that represents that you had initially high expression of this gene, and it will get some higher fluorescence, and vice versa. If you don't have the gene in your sample, it will not be hybridized, and it will not be shown in the data. And there are two things to consider about it. So one is that because this is 24 mer, and well, who knows how long is the human genome? Yeah, and do you think like this 24 mer uniquely represent any gene or not? Well, actually, yeah, it depends. Like any answer in biology, it depends. So there are parts that are like pretty unique, but there are a lot of, but it's not that informative because in particular we have a lot of repeats in the genome. So this is not always one-to-one mapping. So basically what could happen that this 24 mer can be present in many genes. So basically, so that means that you have basically your measurement, your probe is mapped not to one single gene, but to many genes. And well, this leads to some, well, not problems, but challenges in interpretation. So if you see this 24 mer get upregulated, so does it mean that which gene is changed? Yeah. And another thing like about microarrays is it's because they are fluorescent ones. There is usually a cross fluorescence between these probes, so you won't get zero signal. So even if you, for this probe, there is no gene present in your data, in your sample, there will still be like non-zero value because from cross interaction. Okay, so basically now, yeah, and for each gene there can be multiple probes as well, or like this like 45,000. Or actually, I believe this is exactly the same microarray that we did here. No. Yeah, in particular in this geo data set, you can see that this was done with Affymetrix mouse genome for 30 array. So not yeah, but it's kind of the same. So there was some question in Zoom. Yes? No?
Yeah. I wanted to ask, could you repeat, please, why we always have some signal, even if we have no...
because it's optics. So sequence. Basically what happens that you have like one particular spot that is not fluorescent, but the spots that are adjacent to it are fluorescent because they binds to some gene. And basically it gets some cross fluorescence from this because it's not like really divided between probes. So it's very small. So either some size. Yeah, but this microarray is pretty small. Yeah, so it's like everyone is got together, and it's well, it's just photograph. So yeah. So this basically can't be that well represented. Okay, so we talked about probes. So now in this particular microarray we have 45,000 probes, and this ID column here represents probe ID. And in particular, if you work with Affymetrix microarrays, this the probe IDs usually like has a suffix of like 18 from Affymetrix. Okay, so the gene symbols are like just a short, like, memorable name for gene. And one thing that I also would like to mention here is about gene IDs. So we have gene IDs come here, but actually there is like one of the major problems in bioinformatics is like basic identifiers, gene identifiers or like any other identifiers. And the problem is that there are many of them. And for genes, there are multiple, and I'm talking about like mostly mouse and human stuff here. Nowadays there are two major types of IDs. One of them is ensemble, and this ensemble is supported by Ensemble Consortium. and I guess you'll talk about, like, Kostya will talk about it a bit more. So, and they look like ends something something, like ends something something G for genes, and ends something something T for transcripts. And also there are well, previously called Entrez IDs, and now they like more officially called Ncbi gene IDs, and they look like numbers. So here, and well, sometimes I guess it's not that much used anymore, but before it was pretty used. Reflect transcript IDs, and these are IDs that starts from this NM. And the problem is that basically, while for some genes they are pretty well defined, and you have all the genes, like all the IDs, be consistent with each other. Like for example, this gene like actin B, which is like one of household genes, like pretty, most common genes for quality check or whatever. And it was known before, and this has a simple function, and it's like pretty stable. So there is one ensemble gene ID, there are like multiple transcripts for it, and there is one Entrez ID, and there is one gene name, and well, only for one of the transcripts from ensemble database, there is RefSec transcript ID as well, but not for others. Well, and that happens because ensemble database is more comprehensive compared to RefSec database. However, this is not the case, like for all the genes. So in particular, some of the genes gets renamed after some time. So while these gene symbols or gene names are useful because you can remember them, usually don't want to use them in the analysis because they can be deprecated, like renamed or whatever. You can have one gene name and one different protein name, so there are a lot of weird stuff going on. And another thing that happens sometimes is that some of the genes have, so the mapping between gene IDs is not one-to-one. So there is, like you can do an exercise like at home or maybe later next week actually, it would be easier. Just try to find some ensemble genes that have multiple Entrez genes, or vice versa, like find Entrez gene IDs that have multiple ensemble IDs. And yeah, so basically, like remember like what genes, what type of IDs you're working with, and yeah, so which type of IDs is here? Yeah, these are Entrez genes because they look like numbers, and these are like usually what happens. It's pretty easy for microarrays. So basically, overall difference between ensemble and Entrez genes, that Ncbi genes, Entrez genes are more conservative, so they're like protein coding genes mostly, and so they are very stable. However, ensemble database tries to get like a really big and comprehensive view on the human genome, or the human, not only human genome. So there are a lot of weird genes there, and so on. So basically, for example, when you do Rna sequencing data analysis, you usually want to capture, like, and it's actually better to capture on the processing steps, like all the genes that are possible. Like the better annotations you have, the better your results will be. And this means that your results will be in ensemble genes, and we'll look at it tomorrow. And however, for like pathway analysis or like a lot of downstream applications, you usually want to look at the genes and on like that are stable, like protein coding and so on, and they are much better represented by Entrez IDs. So usually, like your data that you have can be done in different types of IDs, like probe IDs actually, and that are kind of gene IDs, like Entrez IDs or ensemble IDs, but usually at some point you want to go to Entrez IDs because they are simpler to work with. Although this is not always the case, but like for some simple analysis, this is usually the way. Yeah, yeah. well, this is basically, actually, so the question was, yeah, how the data is stored in Microarray. So, and actually we'll look at it, but basically it's like just a matrix of numbers. So we have rows corresponds to genes, columns correspond to all samples, and we have some annotations. So actually, we'll further down, we'll look at some like file formats actually for this. There, actually, let's go like for the formats. Let's go to the first omnibus. So here you have like for microarrays, you have these supplementary files called raw, and this is like raw data, and they are usually stored in cell files. And I don't know what this PHP file is. This is like some binary binary format from like Affymetrix or like Lumina. Yeah, but and then like after some analysis, like after some pre-processing, basically you have this matrix of numbers, and you can store it in whatever format you want. And well, there is some particular format to store it in a geo database, but it's not an like standard for whatever. And there are some other file formats, text-based file formats that we'll talk about. But yeah, basically it's just a matrix with numbers and some annotations. And actually it can be stored like an Excel file or whatever. Okay. Yeah. So now you talk about genes, and one of the first things that you usually want to do when you load some public, like not actually both your dataset and especially public ones, is just to check whether the dataset like makes sense. And one of the ways, one of the like most preferred ways, is to check the genes that you believe should be like, somehow regulated or whatever. And for macrophages, and in particular inflammatory macrophages, one of the markers that like we really like this ECOD1 gene, which was called recently, which was renamed a few years ago, and before it was called IRG1. And yeah, but nowadays it's called ECOD1. So let's open it, like let's go to it in front of us. So just print ECOD1 in the search menu here, and I click next, for example. And you will see that, well, it's clearly upregulated in LPS and interferon gamma LPS. Yeah, so it's red here on the right and blue on the left. If you look closer, you will see that, well, by the pretty big numbers here, there is also like not zero numbers in interferon gamma case, but mostly zero, well, relatively speaking, mostly zero numbers at untreated samples. So actually, what we'll do, we'll do another type of looking is we'll do this row profile chart, row profile plot. So let's go to this plot chart, and it will ask it like to select columns. So we have a gene selected. Like let's select samples, and you should see something like this. Yep. Do you see it? Just work. Yeah. And well, let's also like add some labels. Title, for example. Yeah. So basically, here we can now easily see that, well, there is indeed some really big upregulation in LPS and interferon gamma plus LPS samples, but there is also like some upregulation in interferon gamma case, like basically from 0 numbers to like 4K. So there is some weird stuff going on here, and we'll like remember it. So we'll talk a bit about it later. Yeah. And basically, you can see that overall overview, like particular numbers of expressions. And Yeah, one of the things that you should notice that the numbers here are indeed very high. So it goes like from 0 to like 30,000. And that means that the data, well, so-called linear scale, and actually, like to see it a bit better, so we saw like this particular gene, so let's do a plot about of all the genes. So let's go back to Phantasus, select all the genes, like with Control A. There will be a warning that you selected too many rows, and ignore it for now. And change the chart type to box plot. And select group by like either geo-accession or title. Okay. Is it working? Yeah. So basically, we see that overall we have a really high range of expression, changing like from 0 to like 50K, which is a lot. And also, like you see that the distribution here is very like weird. It's concentrated in the bottom, and there are some outliers in the upper part. This is not very nice scale to work with. In particular, a lot of the methods for gene expression analysis and for a lot of just statistical methods were developed to work with data that has normal distribution, like this ball-shaped, which is not the case here. So we have like basically for normal distribution we have pretty low, pretty small tails, and here we have very heavy tails. So a lot of genes has high expression. And for and another way to look at it. So basically, there are two reasons why we don't want this. And one of this is that the data, the methods are developed to work with normal scale data, like to normal noise and so on. And another thing that when you, so the gene expression is not absolute, so it's not like concentrations of like molar per liter or whatever. So the numbers that we have are kind of proportional to what to actual concentrations of the genes, like mRNA of the genes in the sample. And also like and how this is relevant to the function, like actual like amount of protein is another thing. And well, the protein also can have different activity and so on. So how this change, like these numbers here, how they represent actual activity of the gene is far from trivial. And basically what you usually want to look at is this differential regulation, or like when you see some changes. And for these changes, you look at not just like changing from A by some number. What you really usually are looking for is proportional change. So this protein got like twofold in upregulation. So the like fourfold, like tenfold. So you don't deal with absolute, you want like proportional changes. And basically, so what you would like to do right now is to apply like log scale transformation. So coming from this big scale to logarithmic scale, and which as well will change like our models that have product in there to models that are linear and sums. So basically, it's our after log scale transformation, our well, twofold change will be like change by 1, for example, if you do a log-2 transformation. So and this basically gets all the results linear, or like all the like it allows to make like linear assumptions and to work with linear methods. So yeah, let's do the log-2 transformation. So plot this chart. Those just look to, okay, yeah, and redo the plot. It should look like this, right? So now, actually, it looks like normal distribution right now, like more or less, from as much as you can guess from box plot. So basically, you have some heavy part in the middle and well limited tails, which is pretty nice. Yeah. So this is like one of the major things that, like for most of the type of analysis, you want to your data to be log scaled, which is well sometimes, and you should like keep in mind if it was log scaled before you, or you have to do the log scale or whatever. So in particular, in our case, it was not log scale, like it was linear scale, so we did the log scale transformation. Yeah, and another thing that we can notice here, okay, now actually we can discern some differences between samples. And basically you can see that some of the samples have more like higher average or like median value of expression compared to other ones. And like for example, this light blue compared to like more dark blue sample. And now this can happen like from two reasons. One is that indeed I know cell grew a bit, like more the cell like the cell in the sample cells in the sample tool like more active, they grow a bit and have more messenger RNA there, but usually it's not the case, and this is like one of the major assumptions about RNA like gene expression data is that mostly you don't have major changes in your data set. So the changes are like most of the genes stay relatively the same if you like on overall. This is not the case for some of the type of biological data, like for example metagenomics samples. They can be very different, so like one from another. But and because of that, they require different methods. But for like gene expression data, mostly you can assume, mostly you can assume that your genes, like if you have like critical cells, didn't change that much. And now, why this can happen? Like another reason why this difference in the overall expression can happen is that you just had a higher amount of mRNA in the library prep. So we just like, had a bigger drop of the sample and so on. So basically, it's not biological effect, but technical effect. So basically, you had a slightly bigger sample, so and that's why you have overall big higher amount of mRNA. Or like if we talk about like RNA sequencing, I think like most of you are at least familiar with it. So basically, you can have different depths of sequencing for different samples. So if you have higher depths of sequencing, well, of course, you on average will have higher expression. And so what we want to do is we don't want to have this effect like we don't want to have this effect in our data, so that we want to only find biological effects, not the technical ones. So and there is one simple method one of the approaches how to deal with it is so-called quantum normalization, which tries to well one of the like most straightforward approach is just to like shift this distribution a bit so that you have the same average, the same medians. However, also there is some different scales, like should you also like multiply by something or not? So there are some models for it, like how to do it better, like in particular for RNA sequencing data, but for like very simple approach that deals with it, like in general, is the content normalization, which gives you which input is like a set of samples, like a set of values, and it results in all the samples having exactly the same distribution. So, well, of course, it does it in some like with some magic, like and assumptions, but usually it works pretty nice. So let's do this content normalization. And the plot. Okay, now we see that, well, as expected from the method, it indeed returned, it made our samples to be like to have the same overall distribution. So now, if we see that some gene is upregulated in like light blue sample, for example, compared to dark blue, we can more strongly believe that it's indeed like biological effect and not just some technical variation because we had a higher amount of sample in that particular well. Okay. Yeah, and one thing that I would like to mention is so basically now we have our data in log scale, and our samples are comparable with each other, so we have the same distributions. And sometimes when it when what people do incorrectly, they do this log to transformation twice, like just like randomly check the log to button and get it like twice. So make sure that you have this kind of scale on the data. So it's usually it's like from 0 to like 20 or like from 0 to 15, something like this. So if you have numbers like from 0 to 4, this means that like it's not a proper gene expression data. If it's like from 0 to 10,000, it's also like it's in linear scale. So like do check this that you don't didn't do some transformation incorrect. Yeah, and also like in particular in Phantasus, you can do this both transformations in the same step. So just check the log 2 and 1 tile. Yeah. Okay. So now we have this kind of good data set, good comparable samples, like in proper scale. So let's try to deal with another problem. And another problem is that we have multiple probes per gene. For example, let's find this gene Ncbi. Just put ACTB here and click like matches to top button on the right. And you will see that there are 5 probes for this particular gene. And actually you can do the row profile chart as well to look at it better. And so now we have these 5 probes, and actually some of them can be the same oligonucleotides, some can be different. So, and the problem is, well, okay, we see that there is some change, right? But how? Like, first of all, actually, what you can see here, especially in the chart plot, that overall the expression doesn't change. The of Ncbi doesn't change. In particular, in this part, it's pretty pretty flat, and this is good because Ncbi is like again like household gene, which can be used in some analysis as a control, so as a gene that doesn't change. So we see that okay, it's pretty even, so which is good. However, like if we do, when you do like the interpretation and so on, we'd better to have information about gene expression change and not some kind of oligonucleotide like 25 NER change. So and now the question is how to get these 5 probes to one value for this Ncbi gene. And again, for this, there are 2 approaches. One of the like most probably like I don't know, naive or straightforward. And okay, what would be the naive and straightforward approach? Well, we have 5 probes for a gene, and you want to have like 5 rows, and you want to have 1 row. What's your suggestions? Like one row that somehow represent this Ncbi gene. Oh, well, what did it normalize? Yeah, get the average. This is like, yeah, again, naive, but not in a bad way. But yeah, this is like most straightforward way. Let's just average all the values like in samples. So we have 5 measurements, like in one sample, 5 in another, and so on. Just like average them, and this could work fine in theory, but in practice, what happens, in particular, what we can find here is that not all of these probes are equal in their quality. And in particular, what sometimes like is a common scenario is that well, you have some noisy probes, in particular the one that have lower expression. And well, if you average in the noise, so you just you had like good probes, you have a bad probe. Now averaging, you have like an okayish probe. But another way how to deal with it is select like the best probe. And well, there can be different understanding like what the probe is the best. And one of them is just to see on the average expression. So if you have like in here, so we have these 5 probes, and well, 4 of them are more or less similar, but the 5th is the bad one, and the bad one has lower expression. And there are some like biological, like technical reasons for this as well, like in microarrays or Rna sequencing data. So what I usually recommend is just to select one probe that has the highest average expression from these 5. And another thing that why it's usually more convenient is because that if you average, you won't be able to trace your results to one particular probe. And if you select one of them, you'll just you will always understand that okay, this is it will be easy to trace your results to physical like, measurements. So okay, this is the gene, this is particular probe. So okay, yes, and you can now see it, check like is there like any other probes and so on. So like technically, it's much more convenient to have like as well to have one particular probe. So for this, let's go tools collapse. Yeah, there are two ways in Phantasus, like either maximum median probe or maximum mean probe. They do more or less the same, but like the average expression is changed, understood either like as a mean, like actual average value, or like median value. And usually like in the slides I put the median value because it's more robust. In particular, you can do this if your data is not log scaled, but in practice you can use average as well.
Sorry.
Yeah.
Could you please answer? I just can't understand. We kind of have different isoforms or differently spliced forms of gene, yeah? In this case,
Yeah.
5 forms. And what if we have different situations for each gene? For example, if we have one gene differentially expressed, one is a form differentially expressed, and all the other are, say, silenced or something like that. Didn't we just lose the information?
Yes, that's a good question. Actually, it's a pretty hard question. So how do we work with isoforms? And usually, and as I mentioned in the beginning, so today we'll talk about like more crude approach. And usually the isoform change is not the most straightforward thing to do. So usually you're just interested in overall gene change, like gene expression change. And the isoform change require for you to have actual data to support it. So the data should be pretty fine-grained, so that can discern different isoforms. And well, the analysis and interpretation is far from easy. So I believe there will be part about it like in the Costas module next week, how to use isoforms. But yeah, it's pretty hard to, well, not that easy at least, to work with isoforms on microarrays, in particular from this one. We have like 40,000 probes, so this is like a gene level microarray. There are like microarrays that have million probes, and from that you can work with like exon level expression. And well, you can say that, okay, this exon is more expressed than this one. But again, getting from exons to isoform, like to particular isoforms, is not trivial, and so on. So basically, like for this, RNA sequencing data is a better approach, and for this, there are particular methods to assign RNA sequencing reads to isoforms, and like special methods that try to compare them. So, like isoforms is not the easiest way to look at the data, like not the easiest way to understand the data. So basically, with gene expression, which is now became like very common tool in biological research, basically you don't know what to do, just do RNA sequencing like microarray, and it will tell you some gene expression changes and so on. And yeah, in this case, you just usually want to look at genes, and in particular protein coding genes, because again, the genes, you can have 20,000 like protein coding genes, and there are like 40K genes on top, which are like non-coding or whatever, or maybe not genes at all. So usually, for most of the cases, you want to go for genes, for protein coding genes in particular, and well, you do the analysis there, and then if you don't, and then either if you don't see like anything in particular, or if you have, if you did want to look at the isoform change in from the beginning, well, you can try to work with isoforms. But yeah, it requires different design, like experimental design and so on. So basically, nowadays it's maybe better like to work with like long grid sequencing, like nanopore or whatever. So isoform is not that easy.
Thank you.
So yeah, we sometimes lose some information, but basically, again, one of the ideas that we have, we want to for you to understand from this course is that usually, and I guess Nikita Artemov told you this joke that you have some gene from GWAS, and if you can't bet your laptop on it, well, you don't want to do experiment, to make some people do experimental validation, because experimental validation takes cost a lot of money. And again, here usually, at least at the beginning, you want to go for the results that are very pronounced, so and that you can trust completely. And then and then if you don't have these results, you can try to understand what's more fine-grained thing that can go different, but that requires much more support, data support. We should find some other data sets and so on. But yeah, so basically on the gene level, usually, even on the gene level, sometimes you don't trust. Well, it's hard to interpret it and experiment and validate experimentally 100%. So you still have to see what the results will be. But if you look at isoforms, well, more some other non-coding RNAs, it's not easy to go to experimental validation, because no one will believe you, unless you pay for it. Yeah. So basically, the idea is that we want first for bigger changes and more pronounced, more interpretable, gene level and so on. And maybe we'll lose some information there, but it's not usually it's not the problem. Okay. So yeah, let's do a couple of more things and get a small break. Yeah. So now we have we went from 45K genes, 45K probes, to 20K genes, and this is a number of genes, protein coding genes in mouse and human. So this is a number you usually want to see. Again, you can have a lot of non-coding RNAs, but you usually don't want to look at it. You usually don't pursue it in particular. Okay. However, again, as usually you don't want to look at all these 20,000 genes, because some of them are not really expressed in the sample, or maybe even lowly expressed and it's very noisy. So it's usually better to have more robust analysis to just select most expressed genes. And while in RNA sequencing, for example, you can have because actual reads, you can have 0 reads, which means that you can. It's much easier to tell which genes are not expressed. At least if you have 0 reads, probably it's not expressed. However, as I said, in microarrays they have this cross fluorescence, so you don't have 0 values. So it's hard to say if it's there is signal measurements for every probe and for every gene. So, however, there is one simple way that we usually how we can deal with it is just to filter the genes by the average expression and get some kind of top of the highly expressed genes. So for this, let's calculate the average expression of the genes. So let's go to tools, calculation. Yeah, and well, you can sort by this column, and you can see that on if you the genes that are more highly expressed, there are some kind of patterns, either more expressed in the untreated samples or in the treated samples. And if you sort the other way, yeah, so there will be a lot of genes that have expression, average expression around 0, and this is just basically noise. And well, while sometimes noise is not that problem, but usually you do want to remove the noise and just to have the more signal in your data. So yeah. Oh, actually, I forgot to ask. So now, if you look for X and B here, yep, you should see only one match, right? And now again, you can trace this particular value to this particular probe. So this can go back to your data, to the original data, which is good. Yep. So let's go to this filter. We want to filter by this average expression. Well, again, there are two ways to do it. Either like to have a particular threshold for expression, which is not, well, it's not easy to say like which, robustly like which ones you should use. So what we usually do and recommend is to have this top filter. So and select something like 12,000 genes or like 10,000 genes or something like this. And yeah, and this particular number comes from the like empirical observation that actually, and you will also like see it a bit when you work with single cell data sets. That's actually not like per cell there are around 6,000 genes expressed or so, like per each cell, and per each cell type with some roughness. Actually, like 10K is a pretty good estimation on the number of genes that you really want to look at. And well, you can have usually you have just like 12K to have this. Can get more robust, and usually you can get like even more genes if you have diverse data set, like cancer data set, for example, and like big cohort of cancer samples, which are very diverse, and the different genes can be expressed in different samples. But on average, like around like 10 or 12K, or like 15K for this like cancer samples, these are the numbers that we usually use. But again, it's not. It's usually you don't that you're not that interested in the genes that are like on the border. So usually you want you are interested in gene. You don't want to lose the genes that are highly expressed, but that are lowly expressed. Usually it's not the problem if you lose them altogether. If you're looking for a particular gene, you can always look at it in the raw data. But otherwise, to get the overall picture, you don't need them much. Usually. Okay, so now we have these 12,000 genes. So let's create a new heat map with these genes. So because this filter just applied online. So usually you want to have this in a new tab. And yeah, I actually also I believe I forgot to mention that you see that there are some tabs appearing on the upper part. So basically, every time the expression matrix is changed, a new type appears. So like if you messed up with your data set, you can go back and like redo stuff. And yeah, and you can actually like rename. Okay. Yeah, and one more thing before the break, at least before the break, before 5-minute break. Let's save this dataset. So we worked a lot with this to make it more better, more normalized and in proper scale and so on. So let's save it. So let's go to save dataset. And well, you can give some name. So GCT file will be downloaded. And you can try to open it like in any like notepad, open Office Calc or Excel, and see that actually it has a pretty simple structure. So basically, it's a text file with the same information that you saw in Phantasus. So we have this gene expression values, like numbers or annotations, like sample annotations, gene annotations, and so on. So it's like pretty straightforward text format. Okay, so at this point, let's have a short break. Oh. Mm-hmm. Well, let's get back in like in 5 minutes. So this will be 1847, and I think, well, I'll just check if this room will be available after that, but I expect that we'll have some like half an hour at least delay, maybe. Is it okay with you? Yeah. Oh, sorry about this. So yeah, like 5-minute break. hear me? Can you? Is there anyone on the Zoom yet? Or already?
Yeah, we can hear you.
Yeah, good. Okay, let's continue. So basically, now we have this data set, and we have it saved on our laptop, or the computer, and now let's do the following. Let's actually try to load it from it. Yeah. So one wisdom that I would like to tell you as well is, and like everyone doing backups, and did you try to load from your backup at least once to see that it works? Because yeah, sometimes you think that you are doing backups, but actually you can't load it. So yeah, let's open this dataset we just downloaded to see that it works. Yeah, and we are starting, but more or less. Okay, it's more or less worked. So we have the same data set. Yeah. Okay, sorry. Laptop also need a few minute break. Yeah, we came back, and my laptop is back. So let's see what's going on down there. Yeah, so now we have this dataset, and we check that it's more or less, at least, looks like macrophages, activate macrophages. So let's start some more in-depth exploration. And one of the simple way, standard ways to look at the dataset, like gene expression dataset, is to do a principal component analysis plot. So let's do this, and I'll explain what's going on. So let's go tools, plots, so on this plot, first of all, you can see 16 points, like with each point corresponding to one biological sample. And let's do it by color scheme. Yeah. Yeah. So basically, this is a way to represent your, like all your 16 samples, like one of the ways to represent your 16 samples on the plane, on the 2D plane, and basically what we can see here like practically speaking, is that our samples from one condition are grouped together, samples from different conditions are not grouped together, and so on. So it looks like a good representation. Well, except these guys, like the first samples. So you can see that while like in most groups, like in these green groups and dark blue group, like the three samples are very close together, but the first samples are kind of outliers. So that, well, while they kind of close together, but not that much compared to others. Yeah, and remember this the first samples that we saw in the beginning, that there, like when we looked at ECAD1 expression, that the first samples was somewhat different from other. So this actually means that something happens with the first sample. But otherwise, our dataset looks fine. And now to explain how this works, I would like to use Fish's example from actual publication from PubMed. So yeah, and the idea is like, imagine you have, well, I mean, there is some lake or sea, and there are like different fishes, and from them you can measure the different parameters like height, weight, width, like whatever, and you can plot them like one against the other, like again here length versus height, and now the question you can ask is, okay, well, you see that height and length kind of correlate with each other. So basically that means that if you know the length of the fish, you can kind of guess what its height and so on. So, and the question is, well, do you actually need these two numbers to describe the size of the fish? So, and this kind of can be answered with this principal component analysis, which tries to find this axis in your data that would try to explain your data set in like in most compact way, like linear. So basically now we can say that, okay, if you know length, we know the more or less no height. If you know height, we more or less know length, but there is some noise or some variation that we can't really tell. However, if you look at this line along this long shape, so from this we'll have pretty nice understanding of both height and length, and maybe some other parameters as well. So basically this, and this will be like our first component, first main component that will explain whatever it is the data set more in the most part of the data set, which can be explained using the one linear component. And well, from that you can have the second component. So okay, can we describe more of it, and so on. So basically, there are some different applications of this analysis in general, but one of them is to basically to use this like first two components or like first three components to plot your data on the like 2D scale, because the first axis will explain a lot, and second will explain, well, smaller but still a lot, and so on. So basically, the first two axes will represent a big amount, like a large part of variation in your data. So basically, that means that the plot that you PCA plot like for first two components is well represents your data rather well. And yeah, and actually you can do a PCA on the fish as well, like an actual fish. And again, you explain like most of the points using the length and some other points using height. And again, this is like picture from Wikipedia, for whatever reason. Yeah, like it's from French Wikipedia, I guess. Yeah. So again, now this is our plot. So this PC1, first principal component, PC2, second principal component, and then you can select like PC3 and so on. So they will be explaining smaller and smaller part of the variation. So usually, well, if you have diverged data, you can look at the higher principal components, but usually, like even the first two explain like a lot. In particular, if your data set is like mouse one and pretty straightforward one. And also, like maybe some of you do know other ways to represent like high dimensional points. So and the reason here is that instead of looking at 12,000 genes and trying to understand what was the relation, we are looking at like basically we go from this 12,000 dimensional plane, like space, to this 2D space, which is much more easier to like work with and understand, like and interpret. And there can be different ways to do this. How it's called? Sorry, I forgot what. Like down scaling from higher dimensional space to lower dimensional space, like MDS, and you'll see like MDS, or the one that you also will be working with in single level T-SNE and UMAP. So these are different ways to scale down your data points to like plane, for example. But what is great about PCA and why it's very nice approach is that it's linear. And what does it mean? That it means that the axes can be interpretable. And in this case, in our case, I can see that if you look closer, we'll see that the samples, that like the PC1 actually corresponds to LPS response. So from all the samples on the, well, in my case on the left, the samples have LPS stimulation in them, and samples on the right don't have LPS stimulation. And similarly, PC2 corresponds to interferon gamma response. So we have interferon gamma samples on the top and samples without interferon gamma on the bottom. And this is a nice thing to see. So, like in particular, one nice thing to see that the interferon gamma plus LPS also like corresponds well with this. So basically, it's both like LPS and has parts of LPS response and interferon gamma response, which is like makes sense and is a really nice way to check your data. And one of the things that can happen sometimes, people can mislabel samples. And for example, I saw some data sets were mislabeled. And for example, if we would see that like these samples will be labeled as LPS and this will be labeled as interferon gamma plus LPS, you'll see that okay, it doesn't match. So on one side you have interferon gamma going like from bottom to up to the top, and in the other part from the top to bottom. This like doesn't make sense. So well, it can be like maybe not perfectly aligned, but overall you expect interferon gamma response to be in the more or less the same direction, like in both untreated samples and LPS treated samples. So this is like nice way to check that you're biologically like everything went fine. And you can have different experimental designs. For example, one of particular is to have some kind of gene knockout and stimulation. So what you usually expect is that you usually would like to see your untreated sample, like control samples, like well, untreated samples, both wild type and with knockout, to be in this more or less in the same place, so that basically your knockout shouldn't change the baseline, and then like after this knockout, the samples gets activated in more or less the same direction, but one goes like in one way and the other in somewhat other. again, this sort of things is pretty easy to tell from like PCA plot and to reason in terms of PCA plot. So now here we have this, well, everything seems to be labeled fine, like everything's consistent, so which is great. Okay, so this is about PCA. Yeah, and another way to look at the data, also a common way, is to do clustering, like in particular gene clustering. So here we don't have too many samples, so we don't we won't talk. Well, actually, we'll be talking about sample clustering, but in some different sense. So let's see what kind of gene expression pattern, like gene expression clusters there are. So let's go to tools, clustering, k-means. So again, you'll have to put some number, maybe like 16, and I'll talk a bit about it. So, yeah, so after this, like a column should appear like with clusters, and so let's do this tip to window part. Yeah. Okay, everyone has it? Yeah, yeah, no? Good. So now, so what k-means does, it's well, a method of clustering, and here in this case, we are clustering genes based on the expression profiles. So what it does, it tries to put the gene that has the same expression pattern in the same cluster. And basically now, well, we specify the number of clusters that we want to find. So I put 16, so we got 16 clusters here, and yeah, and each cluster, so, because it has similar profile genes with similar profiles. So if you have this overview, this bird eye view on the dataset, we'll see this patterns looking really nice on the heat map. So for example, on the bottom, we have this cluster of genes that are upregulated in interferon gamma plus LPS, well, and a bit and smaller upregulated, lower upregulated in LPS, and not changed between interferon gamma and untreated samples. And well, there are some other clusters as well, but what you also can see is that these first samples that we already were suspicious in our previous analysis, they also are suspicious here. So we see that the interferon gamma plus LPS first sample looks very different. LPS1 is different from the other ones, and actually in the interferon gamma and untreated, there are some maybe not that pronounced, but different as well. So yeah. Yeah, and one thing, two things about this analysis. So then for this algorithm, the number of clusters specified by the user. So this put 16. So this question, like how many clusters do you want to see? So usually it depends on the number of samples. So the higher the number of samples you have, and like more diverse, like higher the amount of biological conditions do you have, the higher the number of clusters you probably want to see. And yeah, but usually, and it depends on like what you're looking for. So like if you just want to look at like, overall how does it look like, how data set looks like, you can the one thing, and if you want for interpretation, for example, to describe these clusters, this is another one. And there is this particular clustering. Well, it's actually called clustering because each gene is clustered in one of these K clusters. So this means that, and actually should see this on this plot, like on the GIF on the right. So this is like a GIF from Wikipedia about how k-means clustering works. And well, for some genes probably shouldn't be like in any clusters. They mean like outliers, noise, or whatever. Some genes maybe you can't really tell like in this part. Well, they formally are like in the red cluster, but actually probably like biologically or whatever, they should be in this bottom cluster. So it's not that perfect, and in particular, it has to assign each gene somewhere. So the clusters can be noisy. So they can be highly pronounced expression patterns and maybe not that pronounced. Another other thing that it's non-deterministic, so it's random-based algorithms. So while overall picture will be the same, but some details can be different from time to time, and from some, like, you can change a bit the scaling, and maybe some will be different. So don't be afraid if some changed, some things have changed. Yeah, and another type of clustering that you can do is hierarchical clustering, and it's usually applied for samples. And let's apply it here. So let's go to tools, clustering, hierarchical clustering, and well, let's keep the default font. Yeah, and so this hierarchical tree will appear, and how this works, like what does it represent? Basically, it tries to find two, in our case, most correlated samples. I guess, so I completed 3 and 4, or maybe it's from gamma 3 and 4. It says that, okay, these are closest samples, let's get them into one branch of the tree. So that will be like our intermediate cluster. So it tries, next it tries to find like what it can join next. So other most closest part, like probably is like completed 3 and 4, and so on. So this, what it results in, basically you have this branch length, and the shorter it is, the more similar the samples are, or the clusters are, and yeah, and the longer they're more different. And what we can see here, like in this data, for this data set, well, like mostly our samples from one condition are clustered together, well, because they're similar to each other, and probably already saw that they kind of group together on PCA plot or whatever, on k-means clustering. However, here you can see that our like first samples again are outliers. So this Untitled one has much higher, like longer branch compared to this to Untitled 2, 3, and 4. In gamma 1, well, maybe not that long, but still much longer than in gamma 2, 3, and 4. And in particular, these two in gamma plus LPS1 and LPS1 samples are actually clustered together and differently from two like groups like LPS group and in gamma plus LPS group. So actually, they are more different from the same group. So the difference between them is higher than the difference between these two groups. Yeah, but overall, it looks like other ways. It looks like a nice data set. And yeah, and the thing is that now we can tell. So we saw it multiple times already that our first samples are outliers, and actually, it's in this case it's pretty simple. So it's we can see that it kind of corresponds to something in the with the dataset metadata, so the like number of samples. So what you can guess probably that these first samples were somewhere maybe close to the border of the well plate, where all the samples were done, and maybe there's some more information happened, or there's maybe some oxygen leaked or whatever. So basically, we have like some kind of batch effect that affected these 4 samples. And at this point, we can tell, okay, this all 4 samples look like outliers. Well, let's remove them from our further analysis. However, sometimes it's not that easy. Sometimes it's also easy. You have a really nice data set, but one sample is really different from any other. There you can usually also say that, okay, this is outlier. But sometimes it's not that definite. So is it outlier? Is it not outlier? So it's not. There is no correct approach, or not formal and correct approach to deal with it. There are some formal approaches, but they have some problems as well. So usually you want to think about it and probably don't remove stuff that you're not sure is outlier. So in this case, we can be pretty sure that this is an outlier. Or sometimes if you're not sure, don't remove it. Yeah, and in our case, so let's actually remove these samples here because we are pretty sure that they are outliers in our case. So let's select them. I select everything except this first sample and get a new heat map. Yeah. So in our cases, we obtained a good data set. It's scaled properly. It's very consistent with biology. It doesn't have any outliers and so on. And we actually will be working with this data set tomorrow. However, sometimes if the outlier is, and in our case, the outliers, while they are pretty, you can see that they are outliers. However, they're not that different. Or remember from PCA plot and from here, they have kind of similar expression to other groups. So, and what it means that we can keep this as it is. However, if we had our data set, our sample, really some really bad outlier, has low RNA-seq coverage or some weird gene expression signatures, so we have to redo this analysis we have to remove them and redo the analysis because remember, in the quantile normalization step, so this quantile normalization, it uses information from all the samples. So again, if our samples is bad, so it will get this bad information spread across all the other samples. But in our case, it's not that needed. Okay, so we have this data set. So let's save it. Let's save it. And now you can go back to this data set without having to redo all the steps. And actually, we'll load this data set tomorrow. So I'm doing some kind of analysis, like different expression analysis and pathway analysis and so on. Yeah, so that's it for today. And to summarize again, when you look at the gene expression data set, there are a couple of things that you should look at, and particularly before doing any analysis. So one is gene expression scale. So it should be usually it should be log scaled, in range 0 to 20 or something. So if it's not this, well, sometimes it can be some weird microarrays. It can be from -5 to 15 or whatever. But the overall should be range like this, not from 0 to 10K or to 100K. You have data should be normalized between the samples. So to remove this effect of just different amount of the sample that went into sample preparation, or into sequencing or to microarray. Yeah, after that, you do want to look at the marker genes. for your particular like biological process. So again, this you have this macrophage activation. So you want to check that that indeed they are activated and consistently with what you would expect. For example, if you have some maybe knockout, you probably, especially genetic knockout, you don't want to see the gene expression, like of this knockout gene expression in the dataset, and so on. So you want to see that the cell type is the one that you are looking at, and so on. Well, and aside from this, so this one requires like biological knowledge and like analysis like PCA and k-means and the hierarchical clustering. It kind of allows you to get an overview of the dataset to understand like if it's good, other samples inside the condition correlate with each other and so on. So this is the thing that you want to look at in your dataset. So in practice, usually, well, these things are, I guess you always should look at, because like it's even if this like analysis was done multiple times in your laboratory, or like, because it's biology, like everything can work like randomly at some point. So you can have these like this DMDM's activation multiple times, but like on the someday you have this like batch effect for the first samples and so on. It's like because of biology, it's always something can happen. So this part you should really check it. Like knockout could maybe didn't work properly or so. Yeah, and you can save the dataset so that you can work with it later. Okay, and again, like tomorrow. So today, and actually, just know that we took a lot of time to on this part. So basically, we didn't do any analysis at all yet, but we only looked at the data, trying to understand if it's good or not, if we can work with it or not. So, and actually, it's indeed one. So there's like in this bioinformatics analysis part, there's a big part to understand. It takes a lot of effort, like understanding that the data set is good, then the simple analysis, and then it takes a lot for interpretation of the data. Okay, so I guess that's it for today. Sorry for taking half hour longer. Yeah, so if you have any questions, I'm ready for to answer them. Are people on the Zoom there yet?
Yeah, we are here.
Yeah, good. There was a question if there are metrics for identifying outliers, and well, people work on this. There are some methods. However, like mostly they're like in statistical sense. So you get like distortion, and then if it's like more than 3 sigma, then it's outlier. However, the problem with biology, it's very weird. So it's not like real. It's not the proper statistic. You have just like 4 samples and 3 samples and 1 outlier. So it's like there are some approaches like in statistical theory, and this is like a big field in statistical theory. However, applied to biological data, it's usually, well, you can't really do something about it. Yeah, one thing also I forgot to mention about the experimental design. So the outliers happen like all the time. So if you like did the analysis, this is experiment like multiple times, the next one you can have outliers. So and so you should account for this possibility. In particular, the question is like how many replicates do you want to have? So in this case, this was really nice analysis experiment. We had 4 replicates. So after the analysis, we like after we removed outliers, we got like still 3 replicates that are very consistent with each other. So this is a great thing. If you have 2 replicates, for example, in one of these, actually you can really tell if it's outlier or not. So and if you can't tell for some reason, you can't really remove it because like it's very hard. Even like with 3 samples, it's hard to do some statistics. But if with 1 sample, you can't really do anything. Oh, almost anything. So remember to, like, if you take part in experimental design part, so remember that you to account for outliers. And, well, like usually like for mice work, like at least like mice, so they are very consistent. Like they are genetically identical, so they are biologically they are very robust. However, well, in some sense, so it's not like human samples that are very different from each other. So you don't really need that much replicates, so like 3 replicates is enough for really powerful analysis. But if you have 3 replicates and one of these outlier, well, it's not that high amount, like not that powerful analysis as well. However, usually 3 replicates is still like gold standard somehow. So basically, still have 2 replicates if there are some problems, which is, well, maybe not that great, but kind of enough. But if you can allow yourself like 4 replicates, it's much better than 3. And after that, maybe it's not that much in these particular types of experiments. Okay. Any other questions? Okay, then that's it for today. Yeah, thanks everyone. And thanks.
Thank you.
Thank you.
Thank you.
Well, I hope to see you tomorrow. And I hope that we'll start on time.